This is a model to represent the prokaryotic replication fork. To begin, we start with gyrase, which is something that will nick the DNA and reduce tension ahead of the replication fork. This is to relieve any supercoiling and make sure that there are no problems when the replication fork reaches that point ahead of time. Next, we have helicase, which is noted by the zipper along our display. This moves along the replication fork and breaks any hydrogen bonds so that the fork can open up. Next, we have the leading strand and the lagging strand. The leading strand is denoted by green because it is built in a continuous form, and the lagging strand is built in discontinuous fragments, also known as Okazaki fragments, which is why we see short segments here. Next, we have primase, which is something that the enzyme creates to make RNA primers. The RNA primase is only needed once along the leading strand and needed continuously along the lagging strand, which is why we may see them in multiple places. As the lagging strand and the leading strand are built, they are both built from the 5' to 3' direction. This means that on this side you have the 3' in the original DNA, and so this new form of DNA strand will be built from 5' on this side to 3' here on the end, and vice versa with this. Because this is 5', the 5' is going to be building away from the replication fork on the lagging strand, and the leading strand will be built into the replication fork. Next, we have DNA polymerase 3. This elongates the DNA, and this works on both sides to do its job and build the DNA sequences. As the Okazaki fragments are being built, they are also alongside single-stranded binding proteins. These bind to single strands of DNA and protect them and stabilize them to prevent any hairpins or any mistakes within the DNA strand. These small primers are removed by DNA polymerase 1. When DNA polymerase 1 removes the primer, it's filling in the gap along the lagging strand and binding the fragments. Next, you will have ligase, which will come in and operate as a sealant on these nicks and fill in the missing sugar phosphate bonds. These strands are working along the fork to create phosphodiesters, which are connecting the sugar phosphate backbone within the new strands of DNA.